Okay, thank you.
Awesome. So just a quick overview for anyone who's watching this on YouTube. This call is between the DPID Working Group, DSci Labs, LACDR, and the Go Fair Foundation. It's going to be split into two main parts, the first of which being an overview of command line interface tooling related to DPID, and then the back half being more administration around DSci Denver and Go Fair Fellow qualified facilitators, Go Fair Foundation qualified facilitators. So with that being said, Eric, do you want to take it away for maybe a quick overview of some of the work that you're doing with the Human Immunome Project?
Yeah, sure, Eric. Thank you. And this, I think, can be very brief. But there's an interest, a very strong interest from this academic group, from this research group, to have identifiers for a very large number of digital twins, essentially. So the idea is, and the project that we have right now funded and we're in the middle of is the following. So the SARS-CoV-2 virus has a protein on the surface called spike, and you may have heard of the famous spike protein. So it's a protein. It's a very long protein. It's 1,200 amino acids. Right? So there's 20 different kinds of amino acids, but depending on their order, you create different proteins. Spike is one particular sequence, has a function, has a structure, is involved in the disease of around COVID. And all through the pandemic, we kept hearing about these variants of spike that were popping up and sort of eluding the immune system, and sometimes causing harm, sometimes not. So there's a lot of interest, not only in this protein, but in like the variants around it, right? There's awareness that, oh, different mutations can come along and have consequences for us, big or small. So what we wanted to do in this project was to really explicitly kind of dive into that mutation space. So what we've done is we started with the original spike protein, and we've generated all of the one-step, we say, missense mutations. Right? So all of the amino acid substitutions for that protein. If you want to think about the math, it's 1,200 positions, and at every position you can mutate in one of 19 different directions. So it's essentially 19 times 1,200 possible one-step variants. That's a lot, but you know, it's like 24,000 or something. You know, it's a lot, but we can deal with it. If you take two-step mutations, right? So you take that first mutation, but now again, at every position, you can mutate again in 19 different directions, but you can do that combinatorially for every sequence. So you end up with an equation that has factorials in it for those two-step mutations, and now things grow really fast. So there's about 300 million two-step mutations. By the time you get to three-step mutations, you're into like 1 trillion possibilities, right?
And this is all theoretical, right? So these variants they don't exist yet. You're sort of like simulating them in the lab.
Some of them do exist, right?
Yeah. Okay.
Yeah, and that's the interesting part. Is it
Yeah.
We actually have testing stations out there or whatever. We have databases where people have seen, in fact, some of these mutations. The other cool thing is that we can go in the laboratory, and our colleagues in Utrecht are the world's expert, actually, at being able to generate arbitrary mutations in spike, and they have a very sophisticated functional assay for that. So they can get kind of a functional readout of these mutations. Yeah. If you want to know more about the structure, that could also be a project. But we thought, well, the first step in this whole thing is at least to try getting a computational structure prediction for each of these one-step and two-step mutations, and as many of the three steps as we can afford.
Right.
So we're talking about in this project minimally 300 million variants of spike, but each variant being potentially really important, right? So
Yep.
The typical computer scientist wants to somehow compress this information, but kind of the approach we're taking here, you really have to have a database of all 300 million because you want to ask interesting questions about patterns that you see in the structure and the function of these proteins. So the relevant issue here, I think, is that we need identifiers for these 300 million and many more to come.
Yeah.
And what we would like to do is build up essentially a kind of a digital twin approach. So each variant is a twin of a real or potentially real sequence, but we might learn a lot of things about that variant. We could run a simulation and say something about its predicted structure. We might do an experiment in the laboratory and actually get back data that might also say something about the structure or the function. We might see it in a testing station in Shanghai or London, and then we want to record that incident. We might learn that this variant is benign and neutral, and you don't see any effect, or that it's severe and causes great harm. We want to know all these things about each and every variant.
Yep.
The claim is that if we build up a data set like this over time, and many different researchers can be contributing to this cloud of digital twin data, that we begin to build up an evolutionary landscape. And we can begin to maybe start mining this information and start saying, well, where would we expect this virus to evolve next, right? Can we begin to anticipate the evolution of these viruses? And certainly we can always go back in time and do retrospective analyses and see that, and that's what we'll do in this project is, can we actually use the structure data that we generate in the computer to say something meaningful about the actual evolution out of Wuhan back in 2020. So that's the project.
That's amazing. That's really fascinating, Eric. My God.
Well, this is sort of a pedestrian view of it. There's actually some really interesting and for me, much more interesting theoretical issues. I want to map the sequence space, map the protein space and be able to say, why do we see these proteins in nature and not other proteins, right? It's a vast space. It's too large to sample in any meaningful way. The number of sequences vastly outnumbers the number of protons in the universe. So there's no way you're even going to instantiate all these things. But we can begin to characterize these landscapes, I think.
Yeah.
But so that's to say, starting now and then into the far future, we need a lot of identifiers for each and every protein sequence that we care about or are interested in. Maybe we come back to this later, Eric, but the approach that we've committed to in this project is through nanopublications. So I think essentially what we're looking for is a way to use decentralized identifiers in a nanopublication context. If that's meaningful, if that has relevance for DeSci, those kind of questions. But I would say in the next couple of months, we will be actively interested in scratching our heads. Where are we going to get these identifiers, guys? How are we going to do that?
So, and I think that there's definitely a lot of potential overlap here, as we've talked about it a couple times before.
Yeah.
The nanopublication approach is interesting to me personally, as a lot of the conversations around fair digital objects are heating up, and there is a case to be made that nanopubs are an already existing implementation of fair digital objects. So combining that with some of the work through IPFS could actually be very interesting, especially whenever you think about the fact that one individual directed acyclic graph can have multiple subcomponents. So from a nanopublication, you could have the provenance, the assertion, and I forget the third aspect, but having all three of those coming back up to a singular content identifier.
Yeah.
And then at the end of it, once you've got all 300 million proteins, possibility to tie that back into however many, I guess, publication style persistent identifiers you think is relevant. So one single DPID can then reference a larger number. And I see where Edward just raised his hand. What are your thoughts on this?
I had two quick questions. First of all, it sounds really cool. Second is like, what's the size of a like single instance? Like, is that like one sequence?
Yeah.
What's the data size?
Yeah. So indeed. So we have options here on how to represent the sequence, but indeed it would be 1,273 single letters, because we have a single letter code for each of the 20 amino acids. So the kind of the most straightforward, let's say least sophisticated representation here is a string of 1,273 letters, and we need an identifier for that thing.
Yeah.
Yeah.
It's like a kilobyte.
Yeah.
For instance, yeah.
Yeah. But we will then link to a data set. If we did the full length, we calculated if we did the full length folding using AlphaFold, from DeepMind, Google, which we're planning to do at our local Dutch supercomputer. If we do the full length protein, the computed structure that's reported back will total 240 terabytes for those 300 million. I think what we're going to do is we're not going to fold the entire protein, but we're going to focus on this 200 amino acid binding domain, just because we're in a small project. So, I don't know if we want to deal at this moment with 240 terabytes, but we can get down to a terabyte in that case. So we would have these kilobytes of sequences, but they might be linking now to terabytes of information.
Yep.
You
And
could
then
probably compress that further. So I would assume that there's some repeats, and you could also reduce the space of like, I don't know how many letters there would be, for example, but you could reduce that down, and so you could encode that probably to save quite a bit of space versus that 1 kilobyte just right off the bat, I think. Just as a note.
And in fact, what's often done in genomics is that you have what they say like a reference genome. So we can use the original spike protein as like a reference, and then every variant would be just a single, position and letter, right? So
yeah, it's like the delta instead of the
43
whole
goes
thing.
from A to B, right?
So I think there's quite a bit of savings that you can do just on the compression of the data, like how you're saying, and there's a couple different techniques that I think that you could potentially represent each one of those without having to take up 200 terabytes of disk, for example.
Of instantiating each sequence. Yeah. So, we can think about how to do this. If we had a reference sequence that we were mapping to, then, we would have to also have, I think we would have to report the reference sequence and then the mutation and the location and the mutation itself. So it's maybe a bit of a question, by the time you start reporting everything you need to reconstruct the sequence, if it might be more minimal to have just the sequence itself. So some simple questions I think we can try to answer, but we have options there to figure that out.
Okay, just one quick
the
question.
second
I
part,
know, yeah, sorry, real quick. I know, Philip, you had your hand raised, but Sina, Edward, were y'all referencing deduplication through IPFS? Was that the concept you were going for?
No, so there's this thing in computer science called Huffman coding, which is how you can do string compression if there's repeats, and then you could also restrict the domain of like instead of like, so ASCII as a domain, for example, like the ASCII characters as a domain require like a certain number of bytes to represent like every single character in the alphabet. But if you could restrict that further, then you could encode it more efficiently. And with string compression, with compression algorithms, you could compress it even further and reduce the amount of size required. But I feel like I may be prematurely optimizing without understanding the full scope of the goal of this project. So I want to leave it there and then
Fair
maybe
enough.
learn a bit more.
Fair enough.
E
So Philip,
varianto.
I see where you had your hand raised. What, because we've skipped over you a couple times. What were you going for?
Yeah, so just clarification question for context for everybody. So Eric, do you intend to make all of that data that you're going to generate publicly available, or is that sensitive data that needs to be stored on a server where you have access control? And then the second question, do you already have a storage provider? I assume it's Surf but I just wanted to clarify.
Yeah. So the happy circumstance here is that in fact everything's going to be open. There's nothing sensitive here. We will use public reporting of variants that are out there. And as for storage, at the moment it will be Surf Sara. There's always questions about a longer term storage on that, but I think Surf is going to work for us for now.
Okay. And it does bring up some interesting questions around, does Surf Sara have IPFS nodes which can accommodate these content identifiers? If not, can we work with them to get that? And if not, if it's something where they may want to take a little bit more time, do we have a backup plan?
Yeah.
Yep. Okay, those are ones that we do need to consider.
So
Well, this is we're gonna sort of we're sending emails out now. We're lining up our compute time, so we're gonna run these things I think in the coming weeks. But they're just data that can be packaged up later.
Yeah, if it's a terabyte, it's not that hard to send if we need to.
Yeah.
Okay, that's fine.
So my experience working with Surf Sara is that they're extremely accommodating for any sort of like user wishes and requests.
Okay.
So if you, Eric, if you would come to them and you would say, well, we need these persistent identifiers, and for that, we need you guys to spin up an IPFS instance.
Yeah.
Chances are that we will be able to work with them. They may I'm not sure if they it depends a little bit on how much work it is for them. I think like small things that don't take a lot of time, they would just do it, part of their customer service.
Yeah.
If it's a bigger project, they may ask for funding. So I don't know like how much effort it would be for them to spin up an IPFS node, but I'm assuming that it's not going to be a lot.
Okay.
Shouldn't be that much.
I had one other question, like tying back into the needs for like how this ties into the nanopublication needs. So my guess is like you want to be able to like attest to a property of a particular sequence or like particular series of mutations, and that's why you kind of want to have an identifier per sequence.
Yeah, so we really want to begin mapping the landscape of protein structures and sequences. So I need to know for every sequence, I need to know its neighbors, and I need to know the structure of those, properties of those neighbors. And so if you can think of sequence space, the space of all proteins, as a regular graph, right? So every node is a sequence, and that node is connected to, n times 19 other nodes. And each of those nodes is connected to n times 19 other nodes. So you have this very high dimensional, very large volume graph. But there's interesting properties then that, even though you might have, 10 to the 500 possible sequences, which is a pretty small number in protein space, you can still navigate from any protein to any other protein in n steps, right? So the space of possible sequences of 1200 amino acids is enormous. It's 20 to the power 1,200. I mean, it's a fantastically large number, but I can go from one sequence to any other sequence in 1,200 steps or less. And that allows me then to, calculate, say, structures along these trajectories. I can begin mapping that enormous space in a linear time frame. And so this hasn't been done before, not really systematically. So that's what we're really kind of after. But what it means is that we really need, every sequence is a coordinate in this space. So we need to uniquely identify that sequence and that coordination, that point, and its relation to everything else.
So I think
Essentially,
it'll
you're
be
trying
pretty
to.
important.
Sorry,
It'll be
go.
pretty important to choose a encoding scheme. And I think there's several out there for proteins. Just a cursory search shows like blossom encoding, which is like a matrix substitution encoding scheme. And these are all just various lossless compression approaches to encoding protein sequences. And I think if you pick one of them, and then you can commit to encoding all the sequences that are produced in this fashion, you just pick an encoding scheme, then creating identifiers for each of these encoded protein sequences, like in turning them to fixed length identifiers, is pretty straightforward with IPFS. You can, create fixed length CIDs, and then you'll have, I think, a pretty good result in terms of creating identifiers for each of these.
And just to say, Eric, it really is up to your lab as to how you want to handle some of these things. If it makes sense to do some of these different encoding, to run encoding on top of any of this information, we're happy to support. It makes sense to have it as a full expanded nano publication. Also happy to support that.
Yeah, we're going to have an internal discussion about, how and when is it appropriate to really nano publish. I think what I would like in any case is to have all the information that's required for a nano publication, but I don't necessarily need it as RDF in a triple store at any particular moment in time. We can store that information in whatever proficient way is available to us. When it comes time to really share this information, then we would consider, how do we do that? And do we expand it all into rich semantic RDF, or do we still make it available through some other kind of, search mechanism that's more efficient?
That makes perfect sense.
Speaking about search, do you have any requirements for querying, returning identifiers for the different sequences, like returning sequences based on their structure internally?
Yeah, so that's part of this discussion, I think, Sina, about the compression issues. So realize too that we happen to be interested in this one particular protein called spike, but there's a lot of proteins out there, and people have made entire careers on other protein sequences. So their view is always going to be, I've got my favorite protein and variants of those. And so at some point, right, my protein will, if I walk far enough away in sequence space, I enter someone else's neighborhood, right? So all these positions are relative. Our point of view is completely relative in this space. It's the same 20 amino acids for every possible protein. It's just different combinations. So I think the requirements might end up being pretty complicated, pretty sophisticated, right? I want to do analyses on variants of spike, but I might be interested in variants that so I may have sequences from observations from patients or whatever, and now I want to align these sequences with ones for which I've already had in the computer and computed structures for. So I might need to do sequence alignments on that. So at some point, I might need to really instantiate each of these sequences so that I can do analyses on them in relation to a bunch of other sequences. So at any particular point in time, I really might need to decompress that information and actually work with it in a bioinformatics mode Although, you know, storing it, yeah, you might want to think about clever ways of storing it to save space.
Yeah,
How
so I think that the issue there for querying is you want to probably be able to load all the sequences into a some sort of database that will allow you to do effectively it's like efficient substring matching, where it's
That'll
like
be important. Yeah.
yeah, so then you can identify, hey, find me all the, you know, the instances that contain this particular sequence that I find important, right?
Yeah, exactly.
Okay.
It's a
And
kind
then
of
do
push
you
that.
anticipate
Yeah.
do you anticipate um other types of queries other than this like substring matching?
Um, if you leave it to biologists, you will be surprised by the number of different kind of questions that'll be coming. Yeah, it'll be I think a lot of creativity will be brought, and there'll be all kinds of interesting questions that I'm probably not going to be able to anticipate at the moment.
Okay. Well,
Yeah.
I guess at the end of the day, what we can do is provide facilitate creating the kind of like corpus of the list of you know of encoded sequences, and then there can be various approaches to actually making those available for querying depending on the application. And I think those could be kind of two separate tasks.
I agree. So I think the storage and the you know the compute or the analysis are really two different phases. And you know, we will do our own analyses in the project. We'll get our deliverables done. But then there will be a longer term question, you know, if there's a platform or, you know, just a nice specification for people to follow if they want to generate and link their data to this, then the questions will follow about, yeah, what's going to be the most efficient way to actually manage the storage and then also the analysis on these very large data sets.
Yeah,
So
and Eric, I think this in a lot of ways ties back into kind of some of what we originally came into the call for, which was mostly the identifiers themselves and the command line interface tooling that can allow Max and Lily to create these identifiers themselves. So
Yeah.
as much as anything, it's thinking through the most efficient way to solve some of these domain specific problems. Because when you're talking about scales like you are, it does matter quite a bit.
Yeah. Yeah.
But it's also trying to figure out how we can help your grad students do that. So maybe this would be a good time for Edward to talk about some of the work that he's doing with different programmatic tooling for both nodes, DPID, and codecs.
Cool.
Sure. I guess that like the reason Sina is asking this a little bit is to be able to like create addresses for each of these individual things. We need to have a way of structuring them in storage. And the way we choose to structure them is going to allow or block certain ways of referring to them. So even though I do agree that like storage for, you know, like doing explorative queries on this doesn't necessarily need to be the same structure that we use to do like single point indexing, because those two needs are quite different. So the way we build stuff up is basically encoding like arbitrary data structures in IPLD and giving a way of not only addressing like the root of this tree, but having a way of going to the root of the tree and then also like address your way down through the tree. So the naive way of doing this would be just to like toss them all into this one structure. And then you would have a single identifier to the root, but that identifier with a path on top is also like a fully like qualified unique identifier in itself. So basically you would just use the property that you have a like defined structure underneath and use that to like derive unique identifiers. And since this is content addressed, this wouldn't change. So like it's not dependent on any like particular, like even if you append to the data set, you will get like a new version of the root, but the address on the old route would still like make sense computationally.
Yeah.
So this could be one way of doing it. Maybe pause for thoughts, I guess.
Would it be the case then that we were talking about, different kinds of different proteins out there? Could we imagine a separate DSI node for every protein? And all of the variants that you would generate for a particular protein could be on that tree for that node.
Yeah, so it makes sense to have some like broader route for each node, I would say. So like maybe one protein is a good measure. Not sure, possibly. But then you could definitely have this like separated into like different nodes. Yes.
So the number of proteins that we're talking about, is it because I'm forgetting, is it like this is going to be, what is the size, the number of proteins that we're talking about here? Is it that 200, is it in the trillions? Is that what we're talking about?
So I think the big fantastic database, which is what DeepMind trained AlphaFold on, I think has over 1 billion protein sequences. So those are just, 1 billion published sequences. Some of them are related, some of them are, or many of them are not at all related to each other. And they have different structures, different functions. They are isolated from different organisms in nature. So yeah, let's say we're talking on the order at the moment for billions of different sequences, and for each sequence, one could launch an investigation creating, again, billions of variants, right? And kind of the question that interests me is, what are the generic features of the landscapes across these billion sequences?
But Eric,
just to clarify, you talked about the like these roots, like the protein, which could then have like amounts of variations underneath. How many of these like roots in this case would we be talking about?
Yeah,
roughly? Yeah.
Well, so I think it can be whatever people are interested in, but there are potentially billions of those roots. Yeah.
And Eric, just to make sure, that's not necessarily where we're starting with, though.
Not at all. With my project manager hat on, I have a project to deliver to Health Holland. We promised 300 million sequences. And, can we build a pipeline that demonstrates some of the things we need to do? At the end of the project, we can always comment on the technology readiness level of this. And I think so long as we identify, good engineering questions that need to be solved and potential solutions for that, I think we're doing okay. That's for the purposes of this project. But of course, as we kind of get into it, we can always think ahead a little bit about anticipating the what things might look like at scale.
Yep.
Right.
So Eric talked about returning to the tooling question. So basically what I'm building up right now is like library-based interaction with nodes. So basically the same feature set that we have in the web app, but you can programmatically like create an update and like change the data and like publish new versions and stuff.
Yeah.
And the scope of these tools, they're separated a little bit into different like baskets of tools, but one kind of concerns the public publishing part.
Yeah.
And one is more like interacting with our backend systems to allow you to like upload data sets and through our dog is going a bit crazy if you're hearing weird sounds.
We're good.
Sorry, I completely lost track. So these are both going to be available as like integration libraries, which you like import in your programming language, and then let's speak to the servers. But also we can do CLI tooling, so like user applications on the command line for doing this interaction, which is, I guess, exactly what you're gonna need if you wanna like hook this up to other computing processes, and something is finished and you put that chunk in the node and then publish it. That would all be possible.
Yeah.
But then again, it's like I think those parts are way easier, like integration wise. I think the data structuring and like what kind of queries we need to support, what's kind of like a viable chunk size for doing this, and kind of all of these trade-offs are going to be super important. And I think that's where we need to focus. I think like tooling part, that's comparatively quite easy, I would say. We can basically accommodate for more or less any like computational way you would like to create these identifiers for it. But the prerequisite for this being easy is that we have like a defined data structure we want to
Got it. Yeah. And the modeling part, I guess, could include some of these little bits and pieces that would be necessary to reconstruct, say, a nano publication, or the FDO when we wanted to. But so I think I understand where this is going. Would the DSI node be let's say located in the surf environment, or would it be different than the surf compute service environment?
So the node would exist without any like place in the traditional sense, I guess, to be vague and abstract. But then, I mean, the data has to live on disk somewhere. And whether or not the node contains, it just contains, the content identifier for it. And whether or not you have to go to a specific place, or whether or not that data is mirrored in many spots, that's really it's like a good property, but that's like not included in how the nodes and the DPID system works. It just tells you how to get to the identifier you need, and then whether or not anyone can deliver that depends on the traditional like data hosting and
Got it. Yeah.
So if two credible parties both hosted that identifier, it could come from either entity.
Yeah. Got it. And the key here is that the identifier doesn't care whether or not you serve it or Philip serves it.
Yep. Got it.
Okay.
So I had one thing I can't like avoid asking. So how much work is it to generate a specific sequence if you have the mutations that you want to do? Because I'm thinking like lazy computation. So basically the link would just be the instructions that you need to run a small piece of code to just generate the thing that you were looking for.
Yeah. So I guess this is related to the earlier conversation we were having. Because if I understand, Edward, what you're asking is that the identifier would be resolving to some code that would then reconstruct the sequence. Is that it? Yeah, and a process.
That's my idea, and I get that this is like just a naive thought, but I'm curious.
Yeah. So I don't think I think we're sort of in new territory. I think for bioinformatics in general, the run-of-the-mill bioinfo that is done out there, the amount of data is never so overwhelming that they care about these questions. I think this is a question that's emerging only in the context of trying to be very systematic at looking at all the variants out there. All of a sudden the numbers get really huge. So you start thinking about ways to store everything. You know, so I don't know if I have an answer here. And I'm not sure. I have a use case in mind yet for the need of this. Like I say, if we have 300 million sequences, I'm not sure we actually have to do compression. If we have 10 to the power of 300 million, then we have to think more clever. But I think given the scale of the project that we have right now, even if we had just the explicit sequence, you know, in a standard file format, it's probably okay. It's going to be less than a terabyte, I think.
Because the reason that I ask is that if you could do this, then the identifier to that program and the sequence of like things you need would be like equivalent to the actual full data. So it wouldn't really be a difference.
Well,
things like wouldn't exist until you ask for them, and then you can like get the result on the fly, kind of. So we wouldn't need to store all of the uninteresting parts, because out of these like 300 billion something, like how large a portion of those things are actually addressed. So this is where I'm thinking about, can we dynamically like build the results if it's asked for, and build like an API that can answer any question about any sequence without actually storing it anywhere?
Yeah. So I think these are very interesting questions. But I don't know if I have an answer for you at the moment. You know, typically people have sequences, they want to do sequence comparisons, or they'll ask about the N terminus of the protein, or they'll ask about the C terminus of the protein. It'll be, I think, a very wide array of arbitrary type questions that will be hard to model, you know, in the first sense. Yeah.
This is where the queuing part becomes interesting again, because if that's the case, we're not just looking for a way of addressing a single instance. Because if we can do that, then it's if that's enough, then it might work. But if you need to do like, well, I want to have this in some sort of relationship to the things I don't know where they are, or like if they exist, or then it becomes a way trickier issue, because then you can't compute it because you don't know what you want.
Indeed. So it may cost you more in the end to do it that way. You know, so you know there may be, it may be viewed as being some level of inefficiency in storing all this information explicitly, but it might save you later on when it comes time to really query and retrieve data and do analyses. I think we will get a better handle on this working with the 300 million. And you know, I'm not an engineer, so my naive approach is to say, well, let's do things very explicit and very simple-minded, and see how that goes. And we will learn, I think, from there, better ways to really do, you know, more sophisticated storage and retrieval and analysis on the data.
At the end of the day, if we want to be able to represent, let's say, 300 million sequences and make those available and have that as a corpus, like an open science data set, and have some reasonable way of you know, cataloging it so that you can refer to each with a unique identifier, that's totally possible with what we have. And then, you know, the reason why I think we're asking the additional questions is because, you know, we want to understand that kind of application specific use cases for, like, you know, the pattern matching and that sort of thing, to see if we can come up with, you know, potentially a more efficient model to store the data. However, if you do want to, you know, put out that entire library and make that available for future research, that's something that we can do today. But I would say that you know, the reason why we're asking further questions is because the model by or the scheme by which we encode these sequences and then make them available with these identifiers, we could potentially create optimizations that would allow for you know, querying and pattern matching use cases to you know, be, you know, more straightforward versus having to load the entire catalog into a separate database. But if your goal is to produce the catalog for further you know, for further annotation of each of those particularly interesting sequences, that's doable with what we have.
Got it. Got it.
Maybe that is a good starting point, is just structuring these for future publication as opposed to future computation. Giving each one of these a persistent identifier as a part of a nano publication so that they can then be disseminated and referenced as needed. Is that more so where you're going with it, Eric?
Well, I think the first application we have in mind is trying to combine very different classes of data, and it seems like something that should be routine these days. You know, as a good scientist, you really want to draw on different data, but it almost never happens. And so that is, you know, given this theoretical sequence, right? And you have this very simple string of amino acids. We can do experimental things in the laboratory to find out information about proteins like that. We can observe them in nature, or we can run computational experiments. And ideally, I think, you know, what's kind of missing right now is a way to bring all that information together in a handy way. Right now, you know, a biophysicist will have their biophysics data somewhere. The bioinformatics person will have their data sequence database somewhere. And so these are really kind of siloed scientific disciplines right now, but that data should be seamless. It should be really interconnected. And I think part of the secret mission that we're on in this project is, you know, can we build a data structure that just allows us to link for every sequence any kind of information we might learn about it. And I think this comes from our background over the years working with nano publications. You know, a nano publication is by definition an assertion, right? It's one statement about the world. It could be true or false, but you say, you know, this protein, you know, has a structure, and you can link to the structure. This protein can have all kinds of other properties. It can have, you know, an occurrence in time and space and organism. So there's lots of things we can say about that sequence. And I think we want to try and figure out a way that we can link all these different assertions that one would want to make about a sequence in a handy way. You know, given the very, and I think modeling that is straightforward in any one case, but the question here is, you know, how can we scale this to sort of arbitrarily large numbers of sequences and information about those sequences?
Basically, what you want to do is create like an some arbitrary data structure holding an opinion or a claim or something, and be able to point that at basically anything else. This kind of ties excellently into what I've been building the last couple of months, which is like let's say like the upcoming like generalization of the DeepID system, where instead of having like this manifest and this file tree and all of the information surrounding it being like part of that same bundle, instead kind of breaking this up into a graph, where like the data tree would be like one node, and then you can have a like, you know, a sequence type of a node, which you could like relate to in basically anything. Like it's hard to describe because it's very abstract, which is kind of what makes it very powerful.
Well, and I would say what it sounds to me like, Edward, is coming closer to an FDO approach. It's really kind of abstracting these things into objects and finding ways to interconnect the objects, you know, with identifiers that are going to resolve, right, reliably to you know, maybe a little bit of information about those things, but certainly their location. So, and that's why, or you know, that's what we think. In hindsight, working with nanopublications, we think the way we're structuring nanopublications now, that they are actually providing an instance of an FDO. And so what we would say is, you know, here's your identifier. It resolves to this little bit of metadata that says, I have an object of type protein sequence, and here's where it's located. And there might be some other information about, you know, who generated it and what time and day, and, you know, some basic info. But then that identifier can be used to link out to a structure that might have been computed with that sequence, or an observation of that sequence in a patient somewhere, right? Or an experiment that someone ran in a laboratory and generated some information about that sequence. And in each of these data elements are self-contained, self-describing, independent objects that you know, can be linked up together. So yeah, it may be that yeah, that we're thinking along the same lines here anyways.
Yeah, so like to me the issue kind of pops up when you think about like how small, like intuitively you really want to have these granules as small as possible. So basically one node, one sequence, and then you can like do everything, and like if just refer to like a list of them if you want to like do more stuff. But there is overhead in this, particularly on the indexing side.
Yeah.
So the kind of dual-edged sword of content addressing is that it's just like nonsensical hashes if you don't know what you're looking for. So you need a way of building up a contextual graph of information that is like searchable and compatible with like what you're thinking about the things.
Yeah.
And being able to like index this, like the overhead here compared to having like a line in a database is, it's not like enormous, but it's definitely like, depending on the data, it's a lot. So like being able to, you kind of lift the issue from like managing the data to managing all of the contexts.
Yeah.
So there are not like solid walls here, but at some point there's going to be some like scalability thing, because like in theory we can build up this graph and have it like infinitely sized. But if no one can actually hold the entire graph of context on their own machine, then it's very hard. Like you can ask any question, but you don't have the data sets to ask the question to kind of figure out where you're going in the end.
But you can request
this is
the data set, and you have a place to request the data set.
Yeah, so the way this kind of ties into like my gut feeling is that you're not going to be able to do this just inside this like abstract graph. You're going to need to have like anchor points, which could be nodes with these like IPLD trees, where you can have like a compacted way of like identifying something in like a larger data set, and then use this like root as the targets for these kind of very generic ways of saying things.
Well, what I do think is interesting from the discussion today, what's kind of sticking in my brain already is, you know, you can go to a database like UniProt, and it's got, I think, 60 million human proteins, you know, and it's a database that's highly curated. So it's very nice. It's a very nice record-based data repository of all human proteins. It's everything we know about human proteins. I could imagine for each of those human proteins, you could set up an IPFS tree of variance. Right. So I don't want, maybe we don't want to build one giant database of all sequences, but we want to use these anchors, you know, kind of the sequences that we all know and love, the things that have captured the attention of biologists for generations. Those are our anchor points. But the new thing here would be to extend for each protein that we know a tree of variance. And I think one thing that, you know, would be interesting, would be very nice as a project. You know, imagine that we just went to UniProt. We have 60 million human proteins there. In a project, a service came by and just generated all of the 1-step, 2-step, 3-step mutations of all human proteins, just to see how stable these molecules are to mutations, and learn something about the evolutionary capabilities inherent in each of these sequences. By doing it this way, you know, you're not sort of overpopulating or polluting that repository with godzillions of sequences, but you would actually have a way to access via DSI nodes all of the variants that people have generated and studied. That to me seems like, if I understand correctly, that might be a nice proposition. Yeah.
Absolutely.
So let them, biologists, continue to use the databases that they love. You know, there's databases for proteins that come from plants and animals and bacteria and fungus and high temperature proteins, and you know, so there's lots of different databases out there, all for special biological interests. But what would be kind of cool is to have kind of a standard way of setting up a DSI node for each protein that could just use, you know, an ensemble of folding algorithms to start generating the evolutionary landscapes of these things. That would be kind of cool.
Eric, with the intention of respecting your time, I think we did hit the 10 a.m. mark. I know you're incredibly busy. I got the news last night. You will be coming to Denver. Really, Zoom? You will be coming to Denver. So perhaps this is a conversation that we can continue in late February to figure it out. But towards the end of this call, it seems like there's a little bit more alignment around publication of some of these sequences and the web of social trust surrounding each of them, as opposed to direct computation, querying, and usage. So this is good progress. I'm glad we had this call. We may have to gloss over the facilitator qualification aspects. And we do have the Telegram now with you, Andre, and I.
Yeah.
So we can coordinate asynchronously there, if that's okay.
Wonderful. Thank you.
I will say that we do have the tooling to support this use case of building these nodes programmatically. So we're happy to help on that end as that comes up.
Yes.
Okay, Sina, that's great. And, you know, all practical details, including, you know, does this cost money? How much does it cost? How do we finance it? These are all things that, you know, would be interesting next step conversations as well. So.
Yep, absolutely. Sounds good. Are we okay to have next steps in Denver?
Yeah, I am.
All right.
That's fine.
Sounds
Thank you.
good.
Thank you.
Thanks, Eric. We appreciate your time.
Yeah, thank you very much, gentlemen. Appreciate it.
All righty. Bye.
Bye-bye.